All right, so today's lab is about osmoregulation, except it's really about osmoregulating and osmoconforming. So let's look at how animals deal with changes in the osmolarity of the water that they're in. So this could be environmental osmolarity or ambient osmolarity. That's the water that's surrounding the animal and the concentration of solutes in it. And we compare that to the concentration of solutes in their extracellular fluids. Now, for an animal with an open circulatory system, like an arthropod or crustacean, it would be hemolymph. For an animal with a closed circulatory system, like a fish, that would be blood osmolarity that we would be looking at. So this line with a slope of 1 is the line of conformity. And so osmoconformers tend to follow this line if you change the ambient osmolarity. Their hemolymph osmolarity follows that. So we'll make this osmoconformers and osmoregulators are animals that maintain a constant hemolymph osmolarity as the ambient osmolarity changes. This doesn't change. So it's a line with a slope of 0. So whenever there's two different patterns of responses of animals, there's typically benefits for doing it one way and benefits and costs for doing it the other way. So what do you think the benefits of being an osmoregulator are? Think about it for a second. Remember, this means they're maintaining homeostasis for the osmotic concentration of their extracellular body fluids. So the benefits are that they can maintain optimal concentrations for enzyme and protein function throughout their body, and that they can survive in a much wider range of ambient osmolarities because it doesn't change their internal osmolarity, so it doesn't expose their enzymes and other proteins to osmolarities that they're not compatible with. But there's a big cost. So let me write down the benefits. And the cost, it takes a lot of energy to maintain their hemolymph osmolarity constant as the external environment or ambient osmolarity changes. So if a conformer moves into the lower concentrations, it's going to be more concentrated inside its body than the water that it's in. Water will flow in and make it swell up and possibly pop if it's an arthropod and it has an exoskeleton. So that's the danger of being an osmoconformer when you move into fresh water. So why study osmoregulating and osmoconforming in animals? Well, it's a subject that's near and dear to my heart. When I was a postdoctoral researcher, we worked on studying how crawfish survive in fresh water. And we'll talk about some of their adaptations in just a minute. Currently, I'm working on a big research project in Apalachicola with blue crabs, looking at how the changing salinity regimes in Apalachicola Bay, as people in Georgia, especially the suburbs of Atlanta, use up excess fresh water and it doesn't get down to the Apalachicola River, so it changes the salinity in the bay. And we're looking at how that affects the distribution of the crabs, the survival of the crabs, and the barnacles that attach to those crabs in the Apalachicola Bay. Now, students often ask, are they called crayfish or crawfish? Well, let me tell you a story about that. I applied for a job at Nicholls State University in Thibodaux, Louisiana a few years ago, quite a few years ago. And I talked about my research with blind cave crayfish and the reduced metabolic rates that they exhibit. And I went through this whole 45-minute talk about my research that I'd already presented at international meetings, and called them crayfish because my major professor was English and she called them crayfish, and everybody in the International Association called them crayfish. And I gave the talk using that term. At the end of the talk, the first question I got was from this really old professor who raised his hand and he said, "You know, down here we call them crawfish." And so I told him, if you offer me the job, I'll call them whatever the hell you want. And they offered me the job, so I've been calling them crawfish ever since.